Dear viewers, now I will be talking on intrinsic and extrinsic autoregulation. Look at the blackboard. I will be very happy if you use your paper. Take out few piece of paper and pen and then start writing. My topic is intrinsic and extrinsic autoregulation of the heart. Do you know this is the heart? We have the four chambers. This is the heart. We have the four chambers. This is the superior vena cava. This is inferior vena cava. ये जो हार्ट है, इस हार्ट की मैंने एक डायग्राम ड्रॉ किया है, जिसमें मैं ये कहना चाह रहा हूं, दिस इज द राइट एट्रियम, दिस इज लेफ्ट एट्रियम, दिस इज राइट वेंट्रिकल and this is left ventricle, and then here we have the different chambers of the heart, starting from the first and important, all are important chamber, right atrium, left atrium, right ventricle and the left ventricle. My topic is intrinsic and extrinsic autoregulation. Means we don't have to stimulate. Heart has sufficient engine, sufficient system by which heart can be excited by its own. Talking about the pacemaker fiber. Pacemaker fiber, this is SA node. This is SA node. This is we have AV node, and then we have AV bundle. Then this is right bundle of his, and this is left bundle of his, and then we have Purkinje fibers. Now SA node actually is the SA node, AV node, Purkinje fiber. These are all pacemaker fiber. Means the fiber which is present in the SA node can generate electrical impulse by its own. We don't have to stimulate SA node. SA node has a capacity to stimulate by itself. That mechanism I will explain here. That is intrinsic regulation. And then this fiber, pacemaker fiber, how this fiber is influenced by the vagus. This is vagus, and then if we talk about sympathetic, we have the center of sympathetic, and then parasympathetic. Vagus is parasympathetic. And the sympathetic. Again, I am going to repeat. We have the pacemaker fiber, SA node, AV node, Purkinje fiber. But actually, if our heart rate becomes in between 75 to 90, in between this, then this heart rate actually is regulated by the activity coming from the AC node. If our heart rate is less than 60 or less than 50, say it is less than 50, then in that case we can say that AC node is not working. At that condition, the AV node has a role to regulate the all the activities of the heart. If AB node is not working, if the heart rate becomes less than 40, mind that अगर हमारी हार्ट रेट 40 के नीचे चला गया, कभी कभी होता भी है, यू कैन गेट द पेशेंट जिसकी हार्ट रेट 40 के नीचे है, तो उस केस में हम कह सकते हैं Purkinje fiber is the fiber which can generate impulse by its own, and the Purkinje fiber is responsible to control the activities of the heart. Systole, diastole, all the activities of the heart is it can be controlled by the Purkinje fibers. Now coming to the intrinsic autoregulation. Here in this diagram, I am showing that whenever there is an involvement of vagus and the involvement of sympathetic, that comes under extrinsic regulation. What is intrinsic regulation? It is that regulation where the system inside the heart is responsible. In the heart itself, there is a sufficient mechanisms, sufficient tissues which can regulate the activity of the heart. That comes under intrinsic autoregulation. And the extrinsic autoregulation, when this intrinsic activity is influenced by the vagus and the sympathetic. As we all know, the vagus and parasympathetic and sympathetic is the component of autonomic nervous system, and our heart is controlled by the autonomic nervous system. So that is why we must look what type of mechanism going on in the heart, both intrinsic and the extrinsic. Coming to the I have drawn here two cells. I have drawn here two cells. One is nodal cell and another is contractile cell. मैं फिर से रिपीट करना चाह रहा हूं. मैंने यहाँ पे जो डायग्राम ड्रॉ किया है, दो सेल का मैं डायग्राम ड्रॉ किया है, एक नोडल सेल और एक कॉन्ट्रैक्टाइल सेल. नोडल सेल मतलब इट इज द सेल व्हिच इज प्रेजेंट इन द एसए नोड, एबी नोड और द Purkinje fiber. The nodal cell means it is the specialized cell, and all these cells मिलके जो बनाते हैं, that is the specialized conducting tissues in the heart. तो फिर से मैं अगर डायग्राम ड्रॉ करूं, this is the heart, and here we have SA node, we have AV node, we have SA node, we have AV node. उसके बाद in between SA node and AV node we have internodal pathways, and then there is another bundle we can see. It is the connector or the tissue which is responsible to carry electrical responses, electrical sensations, electrical activities from SA node towards the left atrium. That is called Bachmann's bundle. Bachmann's bundle. And then AV node. From AV node we have the right bundle, left bundle, and the Purkinje fibers. Now why I am repeating here, जो सेल मैंने ड्रॉ किया है, ये सेल जो है नोडल सेल, means I have taken cell from here, that is the nodal cell, and I have taken one cell from the ventricle or the right atrium, that is the contractile cell. Remember, nodal cell is not involved in contraction and relaxation. We do not have actin myosin component in nodal cell. We have the contractile element in the myocardial cell. So that contractile cell I am talking about, this cell is responsible to contract as soon as there is an electrical stimulus. Now this is the nodal cell. Now this nodal cell is controlled by the sympathetic, and also it can be controlled by the parasympathetic. So here if we talk about, this is vagus and this is parasympathetic, sorry, this is the sympathetic. Now one thing I would like to remind you, the length of preganglionic fiber, here it is long and the postganglionic fiber is short, but here that type of differentiation is not there. मतलब मेरा फिर से मैं रिपीट करना चाह रहा हूँ, यहाँ पे जो प्री गैंग्लियोनिक फाइबर आप देख रहा है, दिस इज गैंग्लियन. जो प्री गैंग्लियोनिक फाइबर है, ये प्री गैंग्लियोनिक फाइबर लंबा होती है और ये शॉर्ट होती है, और ये गैंग्लियन जो है इफेक्टर ऑर्गन में होता है, मीन्स हार्ट के अंदर ये गैंग्लियन मिलेगा. लेकिन सिंपैथेटिक में ये जो प्री गैंग्लियोनिक न्यूरॉन आप देख रहे हैं और पोस्ट गैंग्लियोनिक न्यूरॉन आप देख रहे हैं, ये दोनों न्यूरॉन जो है लेंथ ऑलमोस्ट बराबर की है, मतलब सेम है. देयर इज नो बिगर डिफरेंस बिटवीन द लेंथ बिटवीन द फाइबर्स इफ वी टॉक अबाउट द लेंथ. तो इसके मतलब मेरा ये कहना है, ये जो गैंग्लियन है, दिस गैंग्लियन प्रेजेंट इन बिटवीन हार्ट एंड द स्पाइनल कॉर्ड, बीच में जो है ये गैंग्लियन होता है, हार्ट के ऊपर व हार्ट के अंदर ये नहीं होता है. लेकिन पैरासिम्पैथेटिक का जो गैंग्लियन है वो गैंग्लियन इफेक्टर ऑर्गन में होती है. Beautifully we can do the experiment. If we have the frog, living frog, then beautifully you can stimulate white crescentic line. White crescentic line, actually it is the line, this line is representing the ganglia where preganglion neuron ends and postganglion neuron starts. This white crescentic line present in the heart, and we can stimulate this line easily, and we can see the activity of the heart if we have the frog, and if we can record, if we have the simple kymograph, we can record. Forget about it. Our aim is to talk about intrinsic autoregulation. Now the resting membrane potential here is minus 90 millivolt. Resting membrane potential, sorry, again I will repeat, here it is the minus 60 millivolt. फिर से मैं repeat कर रहा हूँ, जब minus 90 millivolt, here it is a contractile cell जिसकी resting membrane potential minus 90 millivolt होती है, और ये है specialized conducting tissues है जिसकी resting membrane potential जो है minus 60 millivolt. Now coming to the mechanism, intrinsic autoregulation. Here, if we all of you have heard about funny sodium channel. Now funny sodium channel, say this is the diagram of funny sodium channel, and this is the diagram of action potential. What I have said, that the resting membrane potential is minus 60 millivolt, and then this side is millisecond, this is 100, this is 200, and this is 300. This scale is representing the time, millisecond, and this is the millivolt. The resting membrane potential is minus 60 millivolt. Now what will happen? According to the characteristics of this fiber, I am talking about the nodal cell. I am talking about the cell present in the SA node or present in the AB node. Now let us start with the cell which is present in the SA node. The resting membrane potential is minus 60 millivolt. Now here, due to the funny sodium channel, the name of the sodium channel is funny sodium channel. Funny sodium channel. This funny sodium channel, the role of this channel is to bring sodium ion from outside towards inside. So what will happen if the sodium ion is coming in, positive ions are coming, automatically we don't have to stimulate funny sodium channel. The characteristics of this fiber is made such a way that this will bring sodium ions inside the fiber, and that channel we are talking about funny sodium channel. So what will happen? This minus 60 will be brought to minus 45, say 45 millivolt. Now here the threshold potential is minus 40 millivolt. In this fiber, threshold potential is minus 40 millivolt. Now as soon as, due to the action or activity of funny sodium channel, the positive ions will come in. So once positive ion is coming in, then this minus 60 millivolt will be changed to minus 45 millivolt. Say just I am giving one example. मैं एक उदाहरण आपके सामने रख रहा हूँ. तो सोडियम आयन आने से there is a stimulation of T type calcium channel. There are two type of calcium channel. One is T type of calcium channel and another is L type of calcium channel. T is slow and the L is fast channel. Fast channel means T slowly there will be entry of calcium ion. These are all voltage gated channel. So this T channel will be stimulated whenever this volt becomes minus 45 millivolt, means minus 60 millivolt. This minus 60 millivolt is that volt where we can see the activity of funny sodium channel and the sodium ion will come in. Now what will happen? Because of entry of sodium ion, और सोडियम आयन अंदर आता है, अंदर आने से ये जो माइनस 60 मिलीवोल्ट है ये माइनस 45 मिलीवोल्ट हो जाएगी. अगर माइनस 45 मिलीवोल्ट होती है then that millivolt, that amount can be sufficient to stimulate T calcium channel. This is the calcium channel. So there will be an entry of calcium ion. That is the slow channel. That is the voltage gated channel. Two type of calcium channels are there. One T channel, another is L. L is the first channel. This is slow channel. So what will happen? So this potential, this entry of calcium ion here, this voltage now will become minus 40 millivolt. So minus 40 मैंने क्या कहा? Threshold potential. जब ये minus 40 होगी, then what will happen? There will be a stimulation of L channel. There will be a stimulation of L channel, and there will be a huge entry of calcium ion, first channel. So this voltage become plus 40 millivolts. So there will be a huge entry. There will be a huge entry of calcium ion. Now what will happen? Now as soon as this becomes plus 40 millivolt, now as soon as the potential becomes plus 40 millivolt, then there will be a stimulation of potassium channel. So entry of calcium ion will be stopped because voltage become plus 40 millivolt. This is a voltage gated calcium channel. ये voltage gated, voltage के हिसाब से ये काम करेगा. ये तभी काम करेगा जब this becomes minus 40 millivolt. जब ये plus 40 millivolt होगा तब ये काम नहीं करेगा. कि कौन करेगा? पोटासियम आयन जो है, that is also voltage gated potassium channel. So potassium will go out. तो क्या होगा? ये जो potential आप देख रहे हैं, this will come back. So this is called depolarization and this is called repolarization. This is called depolarization. Polarity is gone. Polarity becomes 0 and then it becomes plus 40 millivolt and then again polarity is coming back. So that is why this is called depolarization and this is called repolarization. So total duration becomes near about 250 millisecond to 300 millisecond just to have complete one action potential. If one action potential is there, then we can see the action potential, then we can see this potential also can be changed, can be seen in the contractile cell also. कैसे? Here ये जो gap junction है आपने सुना होगा, we have the gap junction. This is the gap junction. This is the gap junction. Here also we have the gap junction. Please, all of you also at the same time, please draw the diagram. मैंने क्या डायग्राम ड्रॉ किया है? गैप जंक्शन. सेल के बीच में गैप जंक्शन होता है. मैं इसीलिए मैंने आपको ये कहना चाह रहा हूं, जो भी चेंजेस यहाँ पे हो रही है, the same changes we can see here. The same changes we can see here. The minus 90 millivolt. So as soon as there is an entry of sodium ion, funny sodium channel, here also there will be a change with the positive. Then there is a stimulation of slow calcium channel and then fast calcium channel. So this, the resting membrane potential here also the threshold potential becomes minus 70 millivolt. Now due to an entry of positive ion inside the fiber, ये जो पॉजिटिव आयन आप यहाँ पे देख रहा है, द सेम पॉजिटिव आयन यू कैन सी हेयर ऑल्सो. Whatever the potential changes here, the same potential we can see here. Why? Because the pacemaker fiber is attached to the contractile fiber and we have the gap junction. This gap junction will help to bring the potential, to change the potential. What? Minus 90 millivolt. Here also we can see the duration, it is millisecond, and here it is millivolt. Now this minus 90 will be changed to minus, say it is minus 75 and then minus 70 millivolt. So this will come to the threshold potential. As soon as this becomes threshold potential, then there is a not calcium channel. Here the channel is voltage gated sodium channel. So there will be an entry of sodium ion. There is an entry of, so again I will repeat, जैसे यहाँ पे फानी सोडियम चैनल होती है, फानी सोडियम चैनल स्टिमुलेट होती है, तो से क्या होता है, ये जो माइनस 60 मिलीवोल्ट है, बिकॉज ऑफ एंट्री ऑफ पॉजिटिव आयन, दिस बिकम माइनस 45 मिलीवोल्ट, दिस बिकम्स माइनस 40 बिकॉज ऑफ एंट्री ऑफ positive ions, because of entry of sodium ion. Here also the same changes we can see because of the gap junction. So the potential is changed from minus 90 to minus 70. Now instead of stimulating here the calcium voltage gated calcium channel, here there is a stimulation of voltage gated sodium channel. Here also calcium channel is there. I will draw here, calcium channel is also there, but this calcium channel is stimulated whenever the potential is become plus 40 millivolt. तो ये plus 40 कौन लेके आ रहा है? voltage gated sodium channel. जैसे ही there is a threshold potential, means minus 70 millivolt is a threshold potential, then there will be a huge entry of sodium ion, and this huge entry of sodium ion is due to the stimulation of voltage gated sodium channel. Now what will happen? Now there is an entry of calcium channel. There is an entry of calcium ion. So there will be stimulation of calcium channel. That is the L type. And also there is a stimulation of potassium channel, voltage gated potassium channel. So दोनों काम कर रहा है. दोनों काम करने से ये जो पोटेंशियल आप देख रहे हैं plus 40, this is coming down. This becomes say plus 10 millivolt. Plus 10 millivolt coming down. Then what will happen? Then there is a fighting between two. There is a competition between two. If the calcium ion is coming in, ज़्यादा calcium ion आ रहा है, तो we can have the plateau phase. Means the potential will remain plus 10 millivolt for some time. Then what will happen? Then as soon as this becomes plus 10 millivolt, after some time this will be functionless. इसका काम नहीं है. Voltage gated calcium channel will not work. कौन काम करेगा? Potassium channel. There will be an exit of potassium ion. So once there is an exit of potassium ion, then we have this potential, it will come down. Again I will draw this one nicely so that you do not have any problem. So here we have the potential coming down. So here we can see if we look at the different phases, this is the zero phase. This is the zero phase. If you open the textbook, a zero is written there. This is a zero phase. This is first phase. This is second phase. This is first phase. No, I am sorry. This is the first phase. This is the first phase. This is the first phase. This is second phase. This is third phase, and here it is fourth phase. Again I will repeat. फोर फेज मैंने मेंशन किया है. जो डीपोलराइजेशन है, दिस इज जीरो फेज. द पोटेंशियल बिकम्स -90. This will be changed up to +40 millivolt, +40 because of entry of positive ion, sodium ion. Then there is an entry of calcium ion and exit of potassium ion. So because of exit of potassium ion, this will come down. This will come down. So again, if we nicely draw this, so here it is the like, then like, then it is like this. So this is 1, this is 2, this is 3, and this is 4. This 1 means potential is coming down. Because of coming down, +40 changed to +10. It is coming down because of exit of potassium ion and entry of calcium ion. Now this exit will be more than the entry of calcium ion, तो इसीलिए it is coming down. उसके बाद क्या होता है, ये जो सेकेंड फेज दिख रहा है, ये प्लेटिव फेज, इसमें एंट्री ऑफ कैल्शियम आयन ज्यादा होती है फॉर सम टाइम. उसके बाद कैल्शियम आयन जो है इट इज फंक्शनलेस, काम नहीं करते, बंद हो जाती है. तो एग्जिट ऑफ पोटेशियम आयन जो काम करते हैं, तो जो पोटेंशियल जो +10 है, दिस बिकम्स विल बी अगेन -90 मिलीवोल्ट. तो इसको हम कहते हैं रिपोलराइजेशन. फिर थोड़ी देर के लिए ऐसे हमें मिलेगा जिसमें देयर विल बी नो चेंज. जस्ट जो चैनल है, दिस चैनल, स्ट्रक्चर ऑफ द चैनल विल कम बैक टू इट्स नॉर्मल कंडीशन. फिर से आई विल रिपीट, ये जो 4 नंबर की हम बात कर रहे हैं, इसमें फॉर सम टाइम देयर विल बी नो रिस्पॉन्स. द -90 millivolt will remain because in that period, in that time, all these channels will come back, the shape of the channel will come back to normal condition. उसके लिए ये है. उसके बाद फिर से this will come then like this. Again action potential, if there is a generation of action potential here. तो look at this diagram here. अभी भी इंट्रिंसिक ऑटो रेगुलेशन मैंने अभी तो कंप्लीट नहीं हुआ. इंट्रिंसिक ऑटो रेगुलेशन में मुझे ये कहना है कि ये जो पेसमेकर फाइबर है, दिस पेसमेकर फाइबर डज हैव ए कैपेसिटी टू ओरिजिनेट इंपल्स बाय इट्स ओन. इंपल्स मीन एक्शन पोटेंशियल. मैंने आई हैव ड्रॉन द डायग्राम ऑफ एक्शन पोटेंशियल. ड्यूरेशन ऑफ एक्शन पोटेंशियल इज 300 मिलीसेकंड, and in this we have depolarization and we have repolarization. Here there is a fast sodium channel, funny sodium channel. funny sodium channel से क्या होता है, -60 मिलीवोल्ट जो है, दिस बिकम -45. उसके बाद दिस -45 विल स्टिमुलेट द स्लो कैल्शियम चैनल, वोल्टेज गेटेड कैल्शियम चैनल. उसके बाद क्या होगा, ये थ्रेशोल्ड पोटेंशियल में आएगा. थ्रेशोल्ड पोटेंशियल आने से क्या होगा, देयर विल बी ए स्टिमुलेशन ऑफ फर्स्ट एल चैनल, मींस फर्स्ट चैनल, कैल्शियम चैनल. सो दिस पोटेंशियल -45 विल बी चेंज टू +40. उसके बाद क्या होगा, ये बंद हो जाएगा. देयर विल बी एन एग्जिट ऑफ पोटेशियम आयन, तो ये पोटेशियम आयन यहाँ पे हो जाएगा. द सेम चेंजेस हियर, द सेम चेंजेस वी कैन सी हियर आल्सो. देयर विल बी एन एंट्री ऑफ positive ions here entry of positive ion. Now there will be a depolarization and repolarization. The duration here, structurally you can see here there is no plateau phase, but here there is a plateau phase. Means what I want to say is, in this there is a contraction of the fiber. Myocardial cell is contracting here. There will be an entry of calcium ion. There is a formation of actomyosin complex, and I am talking about the contractile fiber. We have actin, myosin. You must have gone through the mechanism of contraction present in the cardiac muscle. I am talking about that. There are 4 stages. One is formation of actomyosin complex. and then joining of a fresh molecule of ATP, then hydrolysis of ATP. These are the steps we can see if you watch my videos where I talked about the mechanism of skeletal muscle contraction, where I mentioned about four molecular steps. First is formation of actomyosin complex, second is power stroke, third is joining of a fresh molecule of ATP, and the fourth is hydrolysis of ATP and formation of energized head. So these are the four mechanisms we can see here, means contraction and relaxation. So contraction is there whenever there is a depolarization, contraction is there, then in this repolarization there is a relaxation. So what I want to say is, in this heart we have enough mechanism. In this heart I am talking about intrinsic, where I am talking about the mechanism which can stimulate heart, which we have the SA node, we have the fiber present in the SA node. This fiber is the specialized conducting tissues of the heart. So there is a generation of action potential here, and this action potential, once it is generated, it will go through the Bachmann's bundle, it will go through the AV node, and then this AV node will carry this action potential and will stimulate the myocardial cell. So myocardial cell we have in the atria, myocardial cell we have in the ventricle. So as soon as there is an action potential, this action potential will be moved through these fibers, through this tract, and then ultimately this will stimulate the myocardial cell. Because of the gap junction, it has a capacity to stimulate, it has a capacity to regulate the myocardial cell. So that is the intrinsic autoregulation of the heart. Again, I request all of you to see my video where I talked about the mechanism which we can see in the heart, means contraction and relaxation. Four steps I have mentioned: formation of actomyosin complex, then power stroke, and then the joining of a fresh molecule of ATP, and the last is hydrolysis of ATP and formation of energized head. So due to that, there is a contraction of the myocardial fiber, and there is a systole and then diastole. So contraction and relaxation, systole and diastole, is again operated by the system which is present in the SA nodal fiber. If SA nodal fiber is not working, then the fiber present in the AV node will work. If AV node is not working, then the Purkinje fiber is there, will control the activity of the heart. Thank you.